Okay, great. So today is the first day of algal bloom monitoring, and today is just introduction. So broadly, we all are sort of dedicated individuals. We are scientists, engineers, and just concerned citizens, and we have come together to form Open Field Collective with two primary agendas. One is to monitor the environmental health around us. And the second is to further science education or inculcate basically scientific temper among the next generation of scientists and change makers. So with that, we all welcome you to our first session today. Broadly, I think what we have today is a global classroom where we are from different countries, the three countries for this pilot program. So I really hope that we can add more dots to this map and create a global, session where we all interact together, come to the same platform, have the same tools in our hands, and learn from one another. Some of us know more about using the tools that we have shared, and some of us know less, but we all sort of interact with one another, irrespective of where we are from and what language we speak. So our vision for this program is twofold. We have a workshop early on, which is what is happening right now. So this workshop will run for about a month. And we'll have 4 sessions, which are primarily educational sessions, where we will explore the idea of what algae is, what algal bloom is, and the equipment that we have shared with you. We'll talk about that. We'll do problem solving together, and just explore the world around us. So that is the workshop part. And then after the workshop, we really hope that all of you will join us in starting a new project called the Algal Bloom Monitoring Project. So it's a long-term project where we all sort of identify a freshwater body around us. And by freshwater body, I mean a lake, it could be a pond, or a river, let's say, any water body that is not connected to sea. And then we go and collect sample once a month, and then do a small experiment for about 1 hour, and then we upload the data on the website. So we all have fun doing that experiment, and it's like a massive project where we all participate together. And using all of that, what we want to create is a global database of freshwater algae. And in that direction, we have already started. And I would like to quickly walk you through one of the pages on our website where we have started collecting a broad database of freshwater algal species with images. So it's kind of very interesting. I will quickly show you. Okay, I need to stop sharing and then share again, because okay, so all of you have probably seen or visited our website. So this is our website, and if you go to the project section and enter the Algal Bloom project, you will see at the very end there is this algal database. So we sort of inherited the database from John Kinross, who is a retired lab technician and a fantastic microscopist in England. So he created this database, and then he kindly agreed to transfer the content on our website, and we plan to expand on what he built. So just quickly, if you go to, let's say, Algal Index, you will find out that the different species have been segregated alphabetically. So if I go to, let's say, C here, and as you can see, all the different kinds of species are named alphabetically. And also what you can find out is images associated with all the scientific names here and a bit of description of those species. So if I click on this, as you can see, I immediately end up looking at a picture where I get some idea of what we are looking at. And if I at all see this in my samples, when I go out into the field, I know what is the scientific name. And that way I can sort of map out what I'm seeing to what exactly it is. So we'll go into the scientific details and we'll talk about the use of the equipment and everything. But today's session is not for that. So today we are just going to say hi to one another and interact with one another. Okay, so with that, I just want to introduce myself. My name is Iqbal, which I should have done before, I know. I totally forgot, but we all are learning. So I'm a researcher, PhD student at Johns Hopkins University, and I'm super excited to work with you all. And I'll open up the platform for everyone to just say a word about who you are and what do you expect from this program, and if why are you with us? That's it. Okay. So on my screen, I'm just going through the list of names that I see first. So Amy, do you want to go ahead?
Hi, everybody. My name is Amy Downs, and I am an elementary teacher by background. And most of my career was spent teaching 5th and 6th grade. I've been a teacher for 28 years. And most recently, I changed grade levels, and now I teach 7th and 8th grade science. So I'm pretty excited about bringing a global community of experts into my classroom. I work at a K-8 school that's really small. Altogether, we have about 430 students. I have about 68 7th and 8th graders that I see every day. And I am from the Central Valley of California. So if you know where, most people don't know where my town is, it's called Visalia. But if you've ever heard of Bakersfield or Fresno, we're kind of in the middle. And if you've never heard of that, we're probably up in the mountains. We're in the valley. Yosemite is about an hour and a half away, but we're in the valley. So thank you for having me be a part of this.
Thank you so much. Ankita?
Hi, I'm Ankita. I'm also associated with Open Field Collective. I'm a postdoc at National Institutes of Health in Bethesda in Maryland in USA. But my origin is from India. I went to France to do my PhD, and then I moved to US. And that sort of makes me also excited about this whole idea about, bringing people together from different countries and doing something, especially around the time of pandemic. So one of the first projects we did was when the pandemic started, because we realized how science education was really important for everybody. And you can pretty much pick any topic or anything you get excited about around you and learn more and more. So because being in science, we are always, curious to learn more and more. So I'm also going to learn along with this group. So my major focus usually is cancer, cancer cell biology and stuff like that. So I know also very little about algae and science involved with that. So I'm pretty excited to learn as well. Thank you. And thank you for joining us.
Thanks, Ankita. Kanika?
Hello, everyone. My name is Kanika, and I was also brought up in India, in Delhi. And right now I am a postdoc at Stanford in California. And I think like all of us here, I'm excited to learn more. And in general, I'm very much excited about microscopy. So during my PhD, I worked on like different tiny microbes, which we cannot see with our naked eyes, but I was studying how they grow and divide using really, really big microscopes, which were like 10, 12 feet high. So, and trying to look at things which are like less than the width of our hair. And that really got me excited about just, like microscopy in general. And then the foldscope was easy one, because in my home, I cannot have a 12 feet microscope, but I can have a small foldscope to look at these things. So yeah, and right now, I'm studying about how bacteria present in our body affect our health and diseases. So I'm excited to learn about a completely different microbe, which are these organisms in water bodies called algae, and we're here to learn together how to see them, how to identify them, so on. Yeah.
Thanks, Kanika. Sayak?
Hi, I'm Sayak. I'm a postdoc at here in Boston at MIT. I'm from Calcutta originally. So, like, unlike the other members of Open Field Collective, I'm not a biologist. So everything that will be taught in this course is also like super new for me as well. So I'm very excited to be a part of this and like actually look at stuff through microscopes. I've never done that so far in my life, so that should be super interesting. Yeah, so I'm really looking forward to it. And like, yeah, what's great about this, having this sort of a global classroom, as Iqbal said, I think it's really amazing. And that's one of the, like, I shouldn't say this, but like one of the pros of the pandemic, that's like so many people can actually sit and interact together, people who would never have originally met. So yeah, so really looking forward to it. Thank you.
Thanks, Sayak. Just to add to that, I have never actually physically met Ankita, Kanika, or Ankit. Right, and we basically have been planning.
Yeah, and we've been working for almost a year or
Yeah,
longer together.
yeah. So we have been working for about a year now, just trying to organize this event. Yeah, we have never met. So that's the weird world we live in right now. So next is Ankit.
Hello, everyone. I'm Ankit Dwivedi. I'm also a postdoctoral researcher working at University of Maryland, Baltimore, here in Maryland, USA. I'm also born and brought up in New Delhi, India. My research background is computational biology and parasites. I specifically work on malaria from using samples from all around the world. This project is very interesting. I mean, I'm very intrigued by this project, not only because I know very less about microscopes or the algae in general, but also I would just want to see how this citizen science approach can answer some questions and how more it can be expanded. And I would thank all of you to kind of being a part of this. And I hope we enjoy our future sessions. Nice meeting you all.
Thank you, Ankit. Next is Dr. D. Snehalata Dodipatti.
Hello, sir. Myself, I am Snehalata. I did my PhD in antimicrobial activity of certain members of freshwater algae. So in this part of this project, I want to learn different forms, identified of different forms in Kakatiya dynastical rivers. I'm very excited to part of this project, sir. Thank you very much.
Thank you so much.
I think we will learn from Snehalata more.
Yeah,
I'm like writing down your name because you actually did PhD on algae.
Yeah, no,
Yeah,
I mean,
madam, just a few, I mean, not all algae, just a few certain members of freshwater algae.
That's awesome.
That's great.
Yeah,
Thank you for joining us.
Thank you, ma'am. Thank you.
Please don't call sir, madam, anybody.
Yeah, please call me Ankit. I was going to say that.
Okay.
Yeah, I mean, I mean, it's a diverse population. Some of us are teachers, some of us are learning PhD students, and some of us, I mean, I think some of the participants are also school students. So yeah, I mean, so you can call us all by our first names, and we all sort of are in the same classroom together.
Okay.
Right?
Thank you.
Next is
It's
Dr. Yeah.
Yeah, okay, that we are very feel free to interact with you. No. Okay.
Thank you so much. Yes. Next is Dr. K. Gunasweta. Gunasweta, you are muted.
Yeah, I'm sorry. I haven't saw that. Once again, I will introduce myself. Hello, everyone. I'm Gunashweta. And presently I'm working in a residential college here in India, where I teach to the degree students. Well, I have completed my PhD on freshwater cyanobacteria as well. And I think throughout my research work, I have isolated around 20 freshwater cyanobacteria, and two have been chosen where I have studied the bioactive nature of those compounds like phenolic compounds and all. So after when I have received the information regarding this project, I was pretty excited because this was somewhere I was connected to my PhD work. So at point I have felt that okay, I think something has been really coming good out of my PhD work, because after a completion of my PhD work, there was no work in India to continue my work on cyanobacteria. So I think this project will be helpful to me, and also it will enhance my understanding towards the algal and their evolution and everything, their studies. So I'm looking forward for this project, and I hope that from me I can teach my students through this project also, that how important it is that studying the our ancient creatures and how they have turned around this entire earth in a useful way. So I think I'm pretty much happy for this. So thank you everyone for making me part of this project. Thank you.
That's awesome. That's awesome, Gunasweta. So do you also want to say a little bit about how many students do you plan to have?
In our college, we have science students around 60 to 70 members. So under me, we have around 90 members. So yeah, because of their 3 years, Indian system is like divided into first year, second year, and third years. So we'll be going for every class, and I think we can actually interact with them. We can teach them. I have my colleague, my head ma'am, the Shulila ma'am, in the project also. We both have been planning this one after we have been selected in this. Like, yeah, this can be also be educational to the students also during the field work, or during the microscopic work. So I think, yeah, we both are pretty much excited for this.
Yeah, that's great. And as you rightly mentioned, cyanobacteria bloom is a major environmental and health risk for, especially during summer season. All right,
Yes, yes.
Moving on. Next is Siva Kumari, Mahendra Hills.
Yeah. Hi, Iqbal.
Hi.
This is Shivkumari from Mahindra Hills. I've just completed my, I'm working as a lecturer in botany in Mahindra Hills, TSWRTC Mahindra Hills. I've just completed my PhD in biohydrogen production and purple non-sulfur bacteria as a biofertilizer. So I'm pretty excited about this project because I usually don't find people working on core botany topics like algae or fungi, and we have very good professors at university, but they are retired. I was just wondering, we have all these integrated sciences, we get a lot of chance to work about the advanced technologies, but when I was just thinking, it's an open collective field and all, and they're working on algal blooms. So I was very pretty excited, and that is why I was happy to take up this, because I want to learn more in the core botany rather than the advanced topics. So I'll be very much happy to do this, because this is the area where I wanted to study actually. And I had I was just thinking to have a take up a project where I can use algae as a biofertilizer, and in continuation to my work that I've done in my PhD, I just want to compare the efficacy of both algae and my own pet microorganism phototrophic bacteria. So this would help me in both ways.
Yeah, I mean, thanks a lot. Algae definitely is an upcoming material for biofertilizer and energy production. I mean, there's a lot of different kinds of application. Yeah, sure. And we are super excited to have you on board.
Yeah.
Thank you.
Thank you.
Next is, let me see, Inian.
Hello.
Hi.
Hi, sir.
Hi, Inian. How are you?
Fine, sir. What?
Can you tell us a little bit about why you wanted to do the project?
We are.
About
I'm
yourself.
studying. Sir,
Go ahead.
I am. We are already practiced in Polscope recently. We want to explore more about water-being organisms in our local areas. That's all.
That sounds wonderful. That's my thing also. I love full scope and I want to explore more.
That's.
Thanks, Inian.
Thank you.
Next is Shiva Leela Kota.
Good evening, everyone.
Yes, we can hear you.
Thank you for joining me. I'm very excited for doing this project, and already Gunashweta ma'am also told that she is a colleague for me, and this is the first project for me. I'm very excited for this. That's it.
Right now, ma'am is saying traveling in train. I think ma'am has a very bad signal right now, so we couldn't hear her.
Thank you for joining us.
properly.
Yeah. All right.
Even from the train.
Thank you, ma'am.
So we have hello. I think your voice is breaking up. So we'll just move on. And next is, let's say, who is next? Sindura Podduturi.
Hello, everyone.
Hi.
This is Sindura, a lecturer in microbiology, and I'm working in TSWRDC Bhopalapalli. And I think it's a great opportunity for me to explore like algal species and looking at them. I think it goes very much crazy. And our college is very much like near to a huge water body, that is Hasanpatti Lake. I often used to think like I would do something related to exploring different bacteria or any other microbes. And this open field collective project has come as a very good opportunity, and I think it would be a great experience for me.
Great. Thank you so much. Uzma Sultana.
Hello everyone, this is Uzma Sultana. I'm a lecturer in TSWRDCW Kamaredi in microbiology. I teach microbiology, and I have done my doctorate in consortia, development of consortia of microorganisms using Pseudomonas, Bacillus, Azoospermillum, and Azotobacter for rain-fed crops like pigeon pea and sorghum. So these rain-fed crops, generally they suffer from abiotic stresses. So actually we have tried for making a consortia, and we have given it in subsidy rates for the poor marginal farmers. This was my doctorate work, and I'm interested as a microbiologist in algae, particularly the pigments produced by the algal species fascinates me towards this work. And also they are eukaryotic photosynthetic life forms which produces their own food materials, and also some of the forms they fix nitrogen with the help of nitrogenase enzyme, and they have a special compartment known as heterocyst. All this excites me a lot, and as Ankita mentioned, I also want to explore fold scope generally. So this has driven me towards this algal bloom project. Thank you.
Thank you so much. Yeah, I mean, we all want to explore something in our surrounding, right? So whether it is algae or tiny organisms or just a leaf or a bee. So we really thank you that you have decided to join us in this journey. Let's go to some younger participants. How about Owen Ko? Owen, are you there?
Hi.
Hi. AON, how are
Hello.
you?
Good.
Do you want to tell us a little bit about yourself?
Well, so I live on a farm. It's right next to a woods, and it has 2 ponds and a creek. So, no, I'm really into nature. So
Oh, and
there's
what?
this one
Yeah,
part
go
of
ahead.
the reason that I took this project is, so nature can be pretty fascinating to me. I've done water testing and in 4-H. And I've also got, and part of the reason why I asked what we could do, because my mom mentioned to me that you couldn't do ponds in this, but when I asked in the chat, somebody said that I could. So, and there's this one, and our 2 ponds have a lot of algae in them, so that'll be fun to figure out what that is. And also how to get rid of it, because I don't think that's good for the pond. But yeah.
That's awesome. Iqbal, you're muted.
Iqbal, you're on mute.
Sorry. Oh, and do you want to tell us a little bit about what grade are you in school?
I am in 7th.
Okay, okay. Next is Kirti Bitlingu.
Yes, sir. Hello, everyone. My name is Kirti. I'm working as a lecturer in botany in Social Welfare Degree College, Adilabad. I did my PhD in plant biotechnology, and I'm very happy to be here. And I like water very much. Like to play and to swim. And at the time of my degree, during my degree, just I collected only few samples for identify algal members, which are surrounded by our Adilabad region. But I played, I swim, everything I did. But I think it is time to collect some samples from such regions and identify and in the microscope. I think this is the time to observe from Adilabad region. And I'm very excited to be here. And thank you so much for giving a great opportunity for me. Thank you.
Great, thank you so much. Let's hear from Lynette. Lynette is in the US and she's an undergrad, I think. Hi, Lynette, you there? We can't hear you.
Museum in LA, and I develop programming for elementary and middle school students. So I'm very interested to learn about how we can kind of use this virtual and kind of field work model and see if I can incorporate it in some of the museum's programming so that we can get local youth out in the field and doing this awesome work. So it's nice to see you all.
Thanks, Lynette.
Thank you for joining us.
Next, shall we go to A. Santosh?
Yes, sir.
Hi.
Hi, sir. I'm Santosh from Tamil Nadu. I'm studying 3rd year UG. I recently worked with Foldscope. I would love to work with the Foldscope. I want to know more about things in water bodies. So I choose. So this is a great opportunity to me. Thanks for the opportunity, sir.
Thanks, Santosh, for joining us. Let's speed it up. Next is Satvik Anantakrishna.
Yeah, hi. So I'm an undergrad, I'm biology undergrad in Chennai, in India, and I wanted to be involved in the project because yeah, I find the Foldscope school cool, and I think I really like to be involved in a citizen science project.
Thanks, Satvik. Next is Jennifer Cyril. Sorry if I mispronounced your last name.
Yeah, that's okay. Hello guys, my name is Jennifer Cyril. I'm from India too. I'm an undergraduate student. I'm currently in my pre-final year. I'm doing my BTech in Industrial Biotechnology from Government College of Technology, Coimbatore. So I joined in this because I wanted to learn more about microbes in the algae. Since my college has been shut down since when I was in my first year, and currently I'm in my pre-final year. So I wanted to learn more, and that's it. I'm looking forward to learn more from you all. Thank you.
Great. Aparna George, you want to tell us a little bit about yourself? Aparna, are you there?
Yeah. Hello, Iqbal. Hello, everyone. Good evening. This is Aparna. I'm working as a microbiology lecturer at Social Welfare Mehboob Nagar College. Yeah, I've never got a chance to work on algae. Though I'm a microbiologist, I didn't get a chance to perform a research on algae. I'm very much interested, and I strongly believe research will help me develop my knowledge and information that drives my students also to know the algal bodies. And I'm very happy to be a part of this global initiative. Thank you all.
Thank you, Aparna. So we have Pavani here. Pavani, are you there?
Yes, Iqbal, I'm very much here, but I'm sorry, I can't enable my video. I've just reached home. There was huge traffic. So hello, everyone, my colleagues and others who have joined from the US as well. I'm really excited to be a part of this program. Actually, I'm just thinking of the day when Somdatta told me about this. Dr. Somdatta is a scientific officer from CCMB. She introduced me to Iqbal, and then from there our conversations went on. We had a meeting, and then today we are having the first session of the program. I'm really, really happy that the project is turning out so well, and I hope the same speed continues when it comes to execution as well. The main idea why we wanted our teachers to be a part of this program is that we wanted our students to benefit from it. As you can see, most of our teachers are already PhDs. They have their PhD from various subjects, botany, zoology, and microbiology. But through them, if the students could learn how to collect the data, how to sort it, how to analyze the data, and contribute their part to whatever science, and then they could do with the minimal equipment that we have received. And also, in that whole process, if they can enjoy learning science and doing science, and that's what I think would be a big achievement for us through this project. So I'm actually hoping that it would happen, and through this project I can see some students getting inspired and taking up science as a career. So I wish the team all the very best. And while you guys get to do all the exciting job of doing science, I'm going to do the boring job of coordinating you all between Iqbal and...
that's the most important thing, trust me.
Yeah, but it's boring, right? You get all the exciting stuff and I get the boring stuff. But
So
yes,
you will also realize, like, during this process, all the members in this step is really important. The people who actually do the work, people who coordinate it, because nothing like this is possible if we don't have all the parts together. So thank you so much. I mean, we're really grateful that you took up, major chunk of this and like transmitting all the information to other teachers. It's like our dream that somebody is actually helping us to do that. So we're very grateful for that.
Thank you, Ankita. That's very nice of you. But anyways, I'm also a scientist by training. I can't call myself a scientist, but I'm a science lover too. I did my PhD in insect immunology, and later I've joined as a lecturer here with our degree colleges, now deputed as a special officer for academics. So that's how I ended up taking this role as a coordinator for this project. So I really wish the team all the best, and I'm super excited. I can't wait to see how this project shapes up as we go through. Yeah. Thank you all.
Thanks, Pavani. Yeah, so Pavani has been, for all of you, Pavani has been instrumental in sort of organizing this event, especially with our participants in India, in the state of Telangana, with the social welfare schools. And yeah, I mean, there were many logistical issues, but Pavani really helped us with that and organizing and identifying the right cohort of students there. So we are also super excited what the students get to learn from you, you all. Okay, moving on, we have next is Sumati Natarajan.
Yeah, hi guys. I'm audible?
Yeah.
Yeah. Yeah, actually, yeah, as Pavani told, I belong to the same group of institutions from India. So it's actually a residential college for women. So most of the women who come here as undergrads for the study are actually coming from a poor background. So if we can actually help them, I mean, in developing a scientific temperament through this project, it would be nothing like it. So I haven't yet tried my hand at algae, though I have done my PhD more involving animal cell culture and signaling. So though I have studied my PG in microbiology, so after PG, not in touch with the freshwater and algae and stuff. So hope to explore that through this. I mean, platform is good. And thank you for including us.
Thank you. Next is, did we already go for Dr. Vasabi Datar?
Hello.
Hi.
Hello, everyone. I'm Dr. Vasavi. I'm also working as a lecturer in microbiology in Telangana Social Welfare Residential Degree College for Women, Mahbub Nagar. So myself and Aparna, both of us thought of taking this opportunity given by you and our Pavani and Joint Secretary. So actually, I have done my PhD on photosynthetic bacteria and hydrogen production. So I'm also excited to take up this, to be active in research. Actually, I wanted to be active in research. So that's why I have joined this project. I also thank all of you for giving me this opportunity. Thank you.
Okay, I think we have, how about next is Sunayana.
Hi, hello everyone. This is Sunaina. I'm also a microbiology lecturer at Social Welfare Degree College, Suryapet. Actually, I'm interested to study the microbial biodiversity in and around our campus of the college. So, with my students, I'm excited to study the algal blooms. In my PhD, I've been working with the photosynthetic bacteria. Now, we are glad to work on the algal blooms. Thank you so much for including me in the group.
Thank you, Sunayana. And I think we have Sandhya Rani left.
She wrote in the chat, so I can read for her if you want. I mean, she just wanted to say hi, and she is excited about this opportunity because she wants to learn more about algae, and she was saying she's coming back from home, so she can switch on her.
Okay.
Thank you, Sandhya, for joining us.
Yeah. So if anyone, so if I missed someone, please just unmute yourself and say hi. I think I have covered everyone, but yeah, did I miss someone? I don't think so.
Hi sir, I am Vijay Lakshmi, lecturer in botany, Mahendra Hills sir. I am very curious about to learn about the microscopic algae. Why? Because most of the photosynthesis occur by algae only. I want to curious to learn about the more and more species of algal members, and whatever I learn, all those things I have to execute to my students also. Thank you very much.
Thank you, Vijaya.
Thank you.
Also, we had Chetna on the chat. She
Yeah.
said hi as well. She's having some network issues. So welcome, Chetna. Thank you for joining us.
Okay. Yeah, I mean, in total, we have around 24 groups, and all groups, like these groups have their own cohort of students, around 5 to 10, I'm assuming. Some of them have more than that. And there are 8 individuals, undergrads and high school students. So yeah, I mean, I'm super excited about what is going to happen, and we'll go through all the details of Algae and Algal Bloom and how to use the equipment. Has everyone received their equipment? Amy, I know that you did not need equipment. You already have those. Yeah, but other than Amy, has everyone received equipment?
Yeah, Iqbal, just a small note. I have asked the teachers to collect the equipment from head office today. I couldn't dispatch it earlier because our secretary, who is the head of the institution, we thought he would interact with the teachers and then we could dispatch the equipment, explain him about the project. But as he was busy, we had to go ahead and tell them only today to collect. So few of them have collected. I think 5 of them have collected. Rest will collect it on Monday.
Yeah, I mean, we have
That sounds
about
perfect,
a week.
yeah.
Yeah, and if you have received your equipment or kit, then it also has a list where you can tally what you have received, and we'll go through that in our next session. We'll share a protocol with you, and we'll share the link to the Algal web that I just went through a bit. Yeah, with that, I just want to say welcome. It has, and please feel free to say unmute yourself and say whatever you want to.
Iqbal, do you want to go through quickly with like the workshop plan we have? I know we sent it on an email, but we could just quickly go through it.
Right.
And if anybody has questions regarding that or would like to include something else,
That's an important point. Yeah. So regarding the workshop plan, so can everyone see my screen?
Yeah, we do.
So I just opened up the email. So this is the plan for the next 4 sessions. So if anyone has any input on how they want to curate it based on their needs or their students' needs, please feel free to chime in and let us know what you want us to explore. So we'll just go over the basics of all that, and we'll sit with you to do interactive live demo sessions and problem solving sessions. But other than that, please let us know where you are coming from and like what else shall we include.
Yeah, so the thing we decided to do also was to go through really the basics. I know a lot of you probably have like more, knowledge than us about microbiology and stuff, but we wanted to go to the basics because a lot of students are still in that process of learning, and we will also learn. So we're just assuming that all of us are on that same level where we don't know anything about anything and we're going to learn step by step. Does that sound good?
Yes, yes.
Okay, perfect. All right. With that, shall we say bye to one another and meet again next
We
week?
look
Same
forward
time?
to see you, next week, same
Yeah.
time, right? Thank
Okay.
you all for joining us. And we'll send you protocols and certain links, so please go through it and maybe, send it to your students as well, whoever is taking care of the cohorts. And if they can view it before and go through it, it'll be wonderful. Thank you all.
Great. Yeah. Thanks, everyone.
Bye.
See you next
Thank you,
week.
Iqbal and Ankita. Thank you so much for having this session. Thank you.
Thank you.
Thank you, Iqbal and Ankita and the whole team. Please, I'll try to join in the next sessions if possible, if I have time.
Sure.
So I'm also eager to learn more about algae. I've never studied algae. In fact, I'm a zoology student. I've been an animal biology student. So it would be interesting to learn something new. Okay, thank you so
same
much.
for me. Yeah.
Yeah. Thank you. We'll see you in the next session. Bye-bye.
Bye.
Bye. chat box is full. I see. I did not see that.
It's okay. We were reading and replying.
Okay.
Yeah, whoever is presenting should not go through the chat box. I mean, the other person
Yeah,
will
you
take
can
care of it. Yeah.
also save the chat to read later.
Yeah, but today's chat, I don't think we have something which it's mostly introduction and stuff. So
I'll still save it.
There are some participants who have not left the chat or the videos.
Maybe we can we remove them?
yeah, Iqbal can because he's the host. Also, is this possible to share like you can stop recording also, Iqbal.
Ah, yes.